Okay, great. So first, thank you so much, Constantine, for inviting me for this very exciting opportunity. It's just really bad that I can't be there in person.
It's a great pity, but we hope that it will happen soon next time.
I really hope so. So here is a picture I downloaded from the internet. I can only imagine myself being in that beautiful location. Um, so as Constantine introduced, I was in the US until 2 months ago, and the work that I'll be presenting today are mostly done at UC Berkeley with the support from HHMI. So I looked at the schedule, and you've had a long morning, and I know that I'm between you and your lunch, so I'll try to be quick. And the other thing is that if you have questions, I don't know how this works, but in case you want to interrupt me and ask questions, I'll be perfectly okay with that. So the topic I'll be presenting today is sort of an outlier. I worked on synaptic plasticity a long time ago, a lifetime ago almost. But in the past 15 years or so, I've been working on sleep. So I'll be talking about sleep. I mean, sleep and working memory and learning are somewhat related, but most of what I'll be talking about today are a little bit far from that, and I apologize for that. So, over the past decade and a half, my lab has been mostly focused on sleep, and we were interested in two fundamental questions about sleep: how we sleep and why we sleep. So sleep is an essential innate behavior. As far as we know, all animals sleep. And that includes the jellyfish, right? So these very simple creatures, they don't even have a brain. They don't have a central nervous system. All they have is sort of a primitive kind of nerve ring that controls some very simple behavior, like some swimming and some feeding behavior. And yet, these guys, they show sleep-like behavior. We know that total sleep deprivation causes death. In fact, we all know that if you're completely food deprived, eventually you will starve to death, right? But in some animals, people have done experiments showing that total sleep deprivation can probably kill an animal faster than food deprivation, just to show you how critical sleep behavior is to the survival of the animal. So for non-mammalian animals, including simple organisms like C. elegans, flies, and fish, sleep is mostly detected by a lack of movement. So for example, for Drosophila, which is used as a very powerful animal model for studying sleep, um, we know we detect sleep when the fly stays still, no movement for at least 5 minutes continuously. And if you apply some gentle sensory stimulation, they should show no motor response. But the readout is completely motor, right? You just observe and see if they move at all. For mammalian animal models, including mice, which is what my lab studies, we usually measure the EEG and the EMG, right? So the EEG measures brain activity, general brain state, and then the EMG measures skeletal muscle tone. It's a measure of somatic motor activity. And you can see that compared to wakefulness, during sleep, the EMG is clearly reduced. And then based on EEG, we can distinguish between REM, rapid eye movement sleep, and non-REM sleep. So you can see that during REM sleep, right, this is when we experience vivid dreams, the EEG looks pretty similar to the wakeful EEG. And this is actually sort of, it has this correlation with conscious activity, right? Because both during dreaming and during wakefulness, you're conscious of something, right? And the EEG shows this sort of high frequency, low amplitude activity. But then during non-REM sleep, this is the deep sleep with very little conscious activity. Then you can see that the EEG is synchronized with a lot of slow but large amplitude activity. So the other thing worth mentioning is that the EEG doesn't distinguish between wakefulness and REM that well, but you can look at the EMG and there's a very clear difference. When you're awake, there's a high muscle tone, but during REM, there's a complete muscle paralysis. In fact, this is called the REM atonia. It's actually a protective mechanism because, when you're dreaming of something dramatic, you might want to act out like, fighting with your enemy or something, but in fact, you're asleep. So this kind of fighting is dangerous. And this kind of REM paralysis can protect us from this kind of, fighting with the imaginary enemy. For humans, if you bring a subject to a sleep clinic, then in addition to EEG and EMG, we'll also measure things like heartbeats and breathing, right? So these also slow down. So these are controlled by the autonomic nervous system. So the point I want to make here is that falling asleep is not just about a change in our brain state or state of consciousness, right? So the brain state you can measure with EEG. It's also about reduction of somatic motor activity that you can measure with the EMG, and also autonomic motor activity, such as the slowdown of heart rate and breathing. Now, when my lab got into the sleep field about 15 years ago, at the time, our main obsession was the question of how do we fall asleep? What's the neuromechanism that controls sleep? And what we discovered over the past decade or so is that the neurons in the mammalian brain that put us to sleep are really part of the motor circuits controlling somatic and autonomic motor activity. So for example, here is a circuit diagram for somatic motor control. Now this blue box here is the basal ganglia, right? And so here you can see the thalamocortical basal ganglia loop. And we all know that this is very important for controlling somatic motor activity. In fact, the basal ganglia is, what goes wrong in Parkinson's disease, and those patients can't move properly, okay? So this is the upper level control motor activity. And of course, below this loop, you also have the midbrain and brainstem nuclei that control some simple motor activity. Now we know that all the brain regions circled in red, they contain neurons that promote sleep. So they can promote, in particular, non-REM sleep. So you can see that it's pretty much every level of this hierarchy. Now, here is a circuit diagram for autonomic control of the cardiovascular system. Right? So this is just a diagram that I found on the internet. And now you can see that we also found sleep neurons in all these regions circled in red. So again, it's pretty much every level of this network. So this led us to propose a motor theory of sleep control, which is that sleep in the mammalian brain, I don't know about flies, my lab doesn't study those, but in the mammalian brain, sleep seems to be controlled by a very distributed network. In fact, probably two networks. One is somatic and the other is autonomic motor network. And so these neurons are basically integral to these two types of motor control. Now, in hindsight, this might even seem obvious, right? Because for mammals and humans, after all, most of the measurements that we use are really measures of motor activity, right? So the EMG is somatic motor activity, and of course, we also have the autonomic motor activity. So most of our measurements, except brain state EEG, right, is about movement. So the fact that a lot of these neurons that put us to sleep are part of the motor circuits might seem even obvious once you know that. But I can tell you that this is not how the field has been thinking about it for almost a century. So today I'm going to show you some of the experiments that we've done that led us to this conclusion. So first, a little bit of a historical background. Pioneered by Marusi and Magoon, who discovered the so-called ascending arousal system. So now we know a lot about the neurons that wake us up. So these include a lot of the neuromodulatory systems. For example, the noradrenergic neurons in the locus coeruleus, cholinergic neurons in both the brainstem and the basal forebrain, some histaminergic neurons, serotonin neurons, dopamine neurons, and so on and so forth, right? So these are quite well characterized brain regions containing these neuromodulatory systems. Pretty much all of them promote wakefulness and arousal. Now, the search for the neuromechanisms controlling sleep really began with the landmark discovery of von Economo. So I'm sure all of us still remember very vividly a few years ago, we came out of this very large flu pandemic, right? COVID-19. Of course, it all affected our lives profoundly. But the biggest flu pandemic in history, recorded in history, actually happened about 100 years ago, right after the First World War. And so some people call it the Spanish flu, right? So that pandemic killed 50 million people worldwide. Is really huge. And what von Economo discovered was that a subset of those patients, right, of that pandemic patients, some of them had severe insomnia. And when he studied the postmortem brains of those patients, he found that those insomnia patients tend to have lesions in this region. It's called the preoptic area, and I'll just call it the POA. So it's part of the anterior hypothalamus. And so this was actually von Economo's original hand drawing. So this red circle indicates the region, the lesion that correlates with insomnia. And then later in animal experiments, people confirmed that if you experimentally lesion this preoptic area, it also causes severe insomnia in animals. So that led to this textbook model with the POA as the sleep center, right? So, think about it. Sleep is this very mysterious process, right? So when you sleep, especially during non-REM sleep, we lose consciousness. Consciousness is such a mysterious thing that's all, so important for all of us. And so it seems to make sense to have this very specialized center that controls sleep, controls the loss of consciousness, right? And of course, we all know that circadian rhythm, which is very closely related to sleep, in the mammalian brain, there's also this master clock in the suprachiasmatic nucleus, right? That circadian center. So it seems that when people found this POA, this idea of a single sleep center made a lot of sense, right? And so it really is written in pretty much all the textbooks. And in fact, this idea had a lot of supports, right? So people, this is just a subset of the papers published on this, and people characterize the sleep promoting neurons in the POA. But it turns out that this wasn't the whole story, because back in the 1950s and 60s, there were several studies that showed that there are probably some neurons in the brainstem, right, very far from the POA. They also have some sleep promoting effects. But then after this burst of papers, over the years, people just kind of forgot about these early studies. But then about a decade ago, Patrick Fuller's lab at a time in Harvard, they found this area, it's called the parafacial zone in the medulla, right, in the hindbrain, that promote non-REM sleep. We also accidentally stumbled upon some midbrain regions with some GABAergic neurons that promote sleep. So all in a sudden, it seems like, this mammalian non-REM sleep is not just controlled by the single center, right? Because there are neurons in the midbrain and also the hindbrain that also seem to promote sleep. So to try to get an idea of what the sleep promoting network might look like, we decided to do a whole brain screening for sleep neurons, right? Because, we accidentally stumbled upon some neurons here and neurons there. But then at some point, you don't want to leave it to accident. So we decided to do a systematic screening for sleep neurons. So we have two basic criteria for sleep neurons. First, they should be sleep promoting, meaning that if you activate these neurons, that should increase sleep. And if you inactivate these neurons, that should decrease sleep. Second, they should be sleep active, right? Meaning that when you're not optogenetically driving these neurons, if you just record their natural activity, they should be active at the right time to promote natural sleep. And when we were doing the whole brain screening for sleep neurons, we also had two corresponding strategies. One is to screen for sleep promoting neurons and then see if they're also sleep active. The second strategy is to screen for sleep active neurons and then see if they're also sleep promoting. So we tried both strategies, and it turns out that both of them worked out much better than I initially thought. In fact, when we started doing this 10 years ago, I thought if just one of them worked to some extent, I'll be very happy. But in fact, both of them work quite well. But today I'll just show you some examples of the first strategy, the sleep promoting neurons. So we did this based on an incredibly simple idea. So we know that sleep and wakefulness are mutually exclusive, right? So if you're asleep, you can't be awake, and if you're awake, you can't be asleep, right? So these are just, by definition, they're not compatible. And so we thought that perhaps we'll just do the following. Now, we know a lot about the neurons that make us wake up, right? So what if we start with those wake promoting neurons, these magenta pink circles, right? And we're going to do retrograde tracing to find neurons in the brain that inhibit these guys, right? Because if you have a neuron that strongly inhibit the neurons that make us wake up, then chances are perhaps that neuron can promote sleep, right? Just because the two states are not compatible with each other. And then once we find these candidate neurons, we're going to do, manipulation and recording to confirm that they are indeed sleep neurons. And once we confirm some of these inhibitory sleep neurons, we're going to trace one more step back and look for their excitatory input. Because if you have a neuron that their activity can cause the activation of other neurons that promote sleep, then perhaps this neuron itself can promote sleep. So this is just an, incredibly simple idea. In fact, I think a smart primary school student can probably come up with an idea like this. So when we first started doing this, I thought this is, way too simple to get us anywhere. But in fact, it took us quite far. So ideas are cheap. And the actual experiments are hard. And so this was actually done by Chen-Yin Ma, a really brilliant former postdoc in my lab. She's actually now my colleague in the Shenzhen in SMART. In fact, her lab is just right next door to my lab. So what Chen-Yin did was to target a whole bunch of wake promoting neurons in the mouse brain. In fact, we know quite a few populations. And she looked for brain regions with broad GABAergic innervation of these wake neurons. So she did this using rabies virus, which allows transsynaptic retrograde tracing. So we really want neurons that we know directly innervate and inhibit these wake neurons. So here is a list of the wake promoting neurons that Chen-Yin targeted. So these include some histaminergic neurons, cholinergic neurons, glutamate and GABAergic neurons, and noradrenergic neurons all over the brain. And here is a list of brain regions with relatively broad GABAergic innervation of these wake neurons. So this list includes the POA, right? So this is the sleep center that people all knew about in the hypothalamus. But to our surprise, the top candidate that came out of this anatomical screening was actually the CEA, the central nucleus of amygdala, which some of you might know is actually involved in learning memory, fear, right? Emotional learning. So Chen-Yin was able to, through a bunch of really smart and creative experiments, she was able to identify a subset of neurons in the CEA that are both sleep active and sleep promoting. But for the sake of time, I'm going to skip this study. It was all published in 2019. And instead, I'll focus on the third candidate region that came out of this anatomical screening. And this is the substantia nigra pars reticulata, the SNR. And this was characterized by another brilliant former postdoc, Dan-Qian Liu, who's now running her own lab in Shanghai, in the institute that Muming Pu founded. I believe you heard his talk a couple of hours ago. So some of you might know that the SNR is actually the output region of the basal ganglia, right? We know that it's very important for controlling movement. So when we did this whole brain screening, we didn't care what each region did, right? All we did was just to do the whole brain screening, right? Based on the simple logic. And yet when we found the SNR, we can't just pretend that we don't know what SNR was all about, right? Because there are all these, hundreds and probably thousands of papers about the role of SNR in movement control. So when we did our experiments, in addition to the standard EEG and EMG, we also added a video recording to look at the motor activity of these mice, just so that we can we can link our findings to the previous literature on the SNR for motor control. So here is a video of the mouse. So when we did this, I believe this was done in 2018 or 2019. It was actually, before AlphaFold, before ChatGPT, DeepSeek, and all these, before AI was so hot, but it was already getting hot. And so we didn't want to be left out. And so we decided to collaborate with our collaborators in Beijing, who helped us to use deep learning to train this convolutional neural net for simple image segmentation. Now, looking back at this study, it was all, like child's play, but I'm really glad that we did this. So this is basically the original mouse, and this is the segmented mouse, and you can see that it's pretty good, right? And so once we were able to automate the image segmentation, it's pretty straightforward to extract some simple parameters for the animal's movement. So when we plotted two parameters, translation and total movement in this two-dimensional plot, we basically saw three clusters. The dark green cluster corresponds to the biggest movement. In this case, is locomotion. So the experiment was all done in the animal's home cage. So the mouse life was pretty boring. Basically, running around is the most exciting thing that he or she did. And then the lighter green cluster is all the smaller movements, including, say, grooming, eating, some postural adjustments, and so forth. So we deliberately did not do the fine-grained classification because we were interested in movement state, not particular movement. So we just lumped all of these smaller movements into one cluster. The third cluster is basically e-mobility, so very little movement. And yet, when we looked at the EEG and EMG, we saw two clusters. One is quiet wakefulness, kind of like what you guys are doing, right? You're awake, hopefully, but you're not moving much. But the other is actual sleep. So basically, we have four behavioral states, right? So 3 wake and 1 sleep states. So here I'm plotting the EEG delta power across the 4 behavioral states, right? So locomotion, smaller movements, quiet wakefulness, and sleep. Now, delta is basically, I think about 0.5 hertz to 4 hertz, more or less, that kind of low frequency range. If you remember the earlier slide that I showed that during non-REM sleep, right, you have a lot of these large amplitude slow activity. So that means high delta. Generally, the higher the delta, the less aroused the brain is. And so here I'm plotting the total EMG power, basically the muscle tone, right? Now, the point I want to make here is that if you look at the 3 kind of greenish states, right, they're all wake states. So for people in the sleep field, they'll just say, oh yeah, when you're awake, it's just one state, right? It's very different from sleep state. But in fact, once you look at the movement, right, even though all 3 are wakeful states, there's also a clear difference in the EEG delta power, meaning that there is a difference in brain state. The more active the body is, the less the delta power, the more aroused the brain is. Here is the EMG power, right? So if you think about quiet wakefulness and non-REM sleep, so both are e-mobility states. So if you study movement, you would say, yeah, that's one state because the animal is not moving. And yet you look at the EMG, there's also a clear difference, right? So when you're really asleep, there's reduction of the muscle tone. In fact, this is why, if you're sitting in a lecture hall and if the talk is too boring, and I'm hoping my talk is not that boring, you nod off, right? When you fall asleep, you basically nod off. And that's because you lose your neck muscle tone, right? So this is a reflection of that change in the muscle tone. So the point here is that brain state measured by EEG and motor state measured by EMG are actually correlated across multiple behavior states. Even during wakefulness, the more your body moves, the more aroused the brain is. Okay, so when we looked at the transitions across the 4 behavior states, they're not random. So if the animal started out in locomotion, dark green, if they transition at all, they only transition into the smaller movements. We have never seen a case where the mouse was like, running around in one moment, the next second he falls asleep, right? Just like we humans, we don't do that, right? When you go to sleep, you don't just like run and then bang, you just fall on the floor and fall asleep, right? Normal humans don't do that, and normal mice don't do that either. And if the mouse started out in smaller movements, they either go back to running or they go into quiet wakefulness. They don't fall asleep right away. So if we actually let me just if we plot these 4 states in a single chain, and then use these arrows to represent the transitions, then most of the transitions are between neighboring states, right? So remember, across these states, there's change in both brain arousal and motor activity. So the fact that most of these transitions are between neighboring states suggests to us that both brain arousal and motor activity tend to change gradually rather than abruptly. Okay, so all of that is just behavior. And what about the neurons, right? Remember, I started out talking about the SNR. So it turns out that there are 2 types of GABAergic neurons in the SNR. The neurons that express parvalbumin, PV, those tend to be sitting in the lateral SNR. And those with a high expression of GAT2, which is a generic marker for GABAergic neurons, they tend to be sitting in the medial SNR. So next, we wanted to look at the activity of each subtype of GABAergic neurons. And for that, we tagged each subtype with a channel rhodopsin. And then we use something called an optrode to record the activity. So basically, this is all homemade. You can train an undergraduate student to make something like this. Basically, it contains an optic fiber surrounded by several bundles of electrodes. You can make these seriotrodes or tetrodes or whatever. So here is an example cell recorded in the GAT2 Cree mice. So these GAT2 neurons were tagged with channel rhodopsin. So you can see that whenever we turn on the laser, this little blue dot for 10 milliseconds, it reliably evoked a spiking, right? So the little black ticks, those represent spikes. And so when we use a high frequency train of laser pulses, it evoked a high frequency train of spikes. And the responses are quite reliable. So once we see a unit like this, we believe that this neuron must express channel rhodopsin. Therefore, in GAT2 Cree mice, they must be GAT2 neurons. And so here is the activity of the cell. So this is the EEG power spectrum, and this is the EMG. And here we're using the color to represent the different behavior states. And this magenta line is the firing rate of this GAT2 neuron. And you can see that this particular neuron has a high firing rate during non-REM sleep, the orange periods, and the activity is lower during either locomotion or other movements. And here's a summary average across the population of GAT2 neurons, this magenta line. So these neurons tend to be most active during sleep and least active during locomotion. But when we record it from the PV neurons, they tend to show the opposite trend. Right? So these neurons tend to be more active during movement and less active during sleep. Okay, so those are just the natural activity of those cells. And what about when we optogenetically manipulate these neurons? So here I'm plotting the so-called laser-triggered average of these different behaviors. So basically, we turn on the laser for 2 minutes per trial. The blue shading here, you can see it's 120 seconds. And so we do this once every 10 minutes or so. And this is the average across all trials from all the mice that we recorded from. So you can see that as soon as we turn on the laser at time 0, there's an immediate drop in local motion and other small movements, and increase in non-REM sleep. And this is for GAT2 neuron activation. And for PV neurons, we saw much weaker effect. So all of this is channel-mediated activation. And when we expressed an inhibitory opson called IC++, and then laser caused the opposite effect, right? So inactivating the GAT2 neurons caused a large increase in movement and decrease in sleep. And again, PV neuron inactivation has a little bit of effect, but the effect is much, much weaker than the GAT2 neurons. So based on both recording and manipulation, we concluded that GAT2, but not the PV neurons, are sleep active, and their activation suppress movement and enhance sleep. Okay, so even though GAT2 neuron activation suppresses movement and enhances sleep, this is not caused by a direct transition from movement to sleep. So these are all the behavior transitions we saw during optogenetic activation with the GAT2 neurons. And you can see that all of these arrows are the ones that we observed under the natural condition without optogenetic manipulation. So what the laser does is to enhance the downward transitions. So all the red arrows are the ones significantly enhanced by laser stimulation, and the blue ones are suppressed, right? So basically what optogenetic activation does is to bias the directions of the natural transitions without creating artificial transitions. Now, again, I just want to sort of stop for a moment and make a comment here. Now, if some of you are a systems neuroscientist like me, right? You're looking for neurons in the brain that control behavior. So if you activate some neurons and you find a transition from locomotion to other movements, you would say, I found these neurons that control movement, right? You would say that these are motor control neurons. And if you activate those neurons and you cause the transition from movement to quiet wakefulness, you would say that's, again, motor control. But if you activate the neurons and you find a transition from quiet wakefulness to sleep, all in a sudden, you don't call these motor neurons, you call these brain state control neurons, because in both cases, there's no change in movement. But in fact, in our case, it's the same population of GAT2 neurons. Their activation causes all these changes, right? In other words, what effect you see does not depend on which neurons you're activating, but depends on where the mouse was. If the mouse was running, your activation causes this transition. If the mouse was in movement, you cause this transition. If they're already in quiet wakefulness, then you cause this transition, right? So in fact, it's the same neurons that can be mediating changes in both motor activity and brain state changes. And when we inactivated these neurons, pretty much it's the same thing, right? So no artificial transitions. All you do is to bias the transitions in the upward direction. So the conclusion here is that basically the activation of these neurons basically cause changes in both motor activity and brain states. It just pushes the animal down the chain, one step at a time. Okay, so next we wanted to look at the downstream targets of the two types of GABAergic neurons. So for the PV neurons, we see no surprise, right? So here we just express some GFP in these cells, and we see the axons in the motor parts of the thalamus, in the motor layers of the superior colliculus. And in this region, people call it the mesencephalic locomotor region, because in the early experiments, people stimulate this region, it causes the animal to run. Right? So in fact, all of these projections, there's nothing new. We're just repeating what people have been saying in the textbooks for a long time. But when we looked at GAT2 neurons, they also project to these motor regions, but in addition, they also project to the dorsal raphe, which contains both serotonin neurons and dopaminergic neurons that are important for regulating brain states. The GAT2 neurons also project to the locus coeruleus, which contains noradrenergic neurons that are important for brain arousal. So in other words, the PV neurons mostly project to the brain motor regions, but the GAT2 neurons project to both the motor regions and the brain arousal regions. And when we use the viral trick to label the subset of GAT2 neurons that project to the motor thalamus, we also saw their axon collaterals in the dorsal raphe and the locus coeruleus. And when we label the subset of neurons that project to the dorsal raphe, we also saw their axon collaterals in these motor regions. So basically, this tells us that it's the same neurons that can send axon collaterals to both the motor regulating regions and also the brain regions regulating brain arousal. And we think that this kind of collateral arrangement allows these neurons to coordinate the changes in brain states and motor states. And as I showed a few slides earlier, pretty much all of these behavioral changes involve both the brain state change and the motor change. Okay, so now we have characterized the population of GABAergic neurons. And as promised before, right, so we're going to trace one more step back and look for their excitatory input. Now, the strongest glutamatergic inputs to the SNR neurons came from this region called the subthalamic nucleus, the STN. So in fact, this is also just textbook stuff, right? So people studying the basal ganglia, they've known for a long time that a major glutamatergic input to SNR neurons came from the STN. And when we activated the STN neurons, in this case, we used neurotensin Cree mice because there are other glutamatergic neurons nearby. So neurotensin is specific. So when we activated these neurons in the STN, we also saw an increase in non-REM sleep and decrease in wakefulness. So that confirmed that the upstream excitatory inputs also promote sleep. Okay, so as I mentioned at the very beginning, right? For humans, we detect sleep based on brain state measurement, somatic motor activity, and autonomic motor activity. So I just told you about the SNR involved in somatic motor activity regulation. So now I'm going to spend a couple minutes to tell you another study on autonomic regulation. So this is the cardiovascular baroreflex circuit, and this was characterized by another really talented former postdoc, Yuanyuan Yao, who's also my colleague now in Shenzhen. So baroreflex is basically a very rapid negative feedback loop that helps us to stabilize our blood pressure. Right? So the blood pressure is one of those things that you don't want to change over a very large range because you might, have a stroke if it goes too high, right? And if it goes too low, then you might faint, right? So how is this stabilized? So the baroreflex circuit goes like the following. There are these baroreceptors sitting in parts of the artery, right? So when blood pressure goes up, these receptors are activated, and then that signal is sent to the brainstem, the medulla region called the nucleus of the solitary tract. And that triggers a number of changes in the heart and the blood vessels to stabilize the blood pressure. So baroreflex has been characterized a long time ago, and there are two main circuits. It starts in the solitary tract. So in one pathway shown on the left, the solitary tract neurons send glutamatergic inputs to this region called the ambiguous nucleus. So the ambiguous nucleus contains cholinergic neurons that are part of the parasympathetic system. So these are the preganglionic parasympathetic neurons, cholinergic. In the second pathway, the glutamatergic projections to the region called the CVLM, caudal ventral lateral medulla. So this region contains GABAergic neurons. These GABAergic neurons project to the RVLM, rostral ventral lateral medulla. So the RVLM contains adrenergic neurons that are part of the sympathetic nervous system, right? So if you think about the logic here, once the solitary tract neurons are activated, it activates the parasympathetic system in this branch, and it disynaptically inhibit the sympathetic pathway. Okay? So in both cases, it sort of promotes the parasympathetic and suppresses sympathetic, therefore to downregulate the blood pressure. So the first thing that Yuan Yuan did was to identify the subset of neurons in the solitary tract that are cardiovascular, because the solitary tract is really the gateway sensing all the visceral internal inputs from the guts, from the lungs, from the heart, and everything, right? So it contains neurons responsible for many different functions. So only a subset are responsible for the cardiovascular system. So Yuan Yuan wanted to identify that subset. So what we did was to use the trap technique developed by Lu Chunluo's lab at Stanford. So basically, we cross two mouse lines together. One mouse line expresses CRE-ER under the FOS promoters. Many of you probably know that FOS is an activity-dependent immediate early gene. So when a neuron is activated, then it turns on the FOS gene. So in this mouse, it would turn on CRE-ER, right? And then this ER is tamoxifen dependent. So if we activate these neurons and then quickly inject tamoxifen, is going to cause CRE-ER to enter the nucleus and then activate this reporter gene. So in this case, we injected phenylephrine, which is a drug that acts on smooth muscles in blood vessels to increase blood pressure. So you can see that after we inject PE, it increased the blood pressure and decreased the heart rate. So it basically kicks off the baroreflex. And if we pair PE injection with this fast-acting tamoxifen injection, then we can turn these neurons on to express CD tomato. So this is the PE trap, and you can see a lot of trap neurons compared to saline control. Okay. So then the next thing that Yuan Yuan did was to see if these PE trap neurons are indeed cardiovascular. So what she did was to tag these neurons with channelrhodopsin, and again use output recording that I showed a few slides earlier to record these activity. So here is an example, solitary tract neuron. And so this was a recording with the mouse kind of, just running around doing whatever he wanted in the home cage. And you can see that depending on the behavior, the blood pressure and the heart rate was going up and then down and up and so forth. And here's a firing rate of solitary tract neuron. And you can see that it follows the blood pressure fluctuation quite well. So this is the recording over, multiple seconds, right? And if we zoom in temporally, it turns out that the subset of these trap neurons are actually time locked to individual heartbeats, right? So here's this ECG. You can see the individual heartbeats. And here is a spike of one of those neurons. So here's the cross correlation between the firing rate of the neuron and the heartbeat, right? So you can see that this is over hundreds of millisecond time scale. And so this very strong correlation means that these neurons are really time locked to individual heartbeats. And we think that that means that these neurons are actually sensitive to blood pressure fluctuations on the time scale of individual heartbeat. Okay, so everything shown in this slide is just to show that these neurons that we trapped are indeed cardiovascular. So what happens if we optogenetically activate these cells? So this shows our standard experiment. In fact, this is how we did experiment in all the earlier studies that I showed you as well. So we basically turn on the laser for 2 minutes, the blue shading here, every 10 or 15 minutes or so with a semi-random kind of sequence. So here is one example period, and this is the summary of all the laser trials from all the mice that we tested. So you can see that when we turn on the laser to activate these trap neurons, it caused an immediate drop in blood pressure and heart rate. So this drop is incredibly fast, over a couple of seconds. But in addition, we also saw a slow but consistent increase in non-REM sleep. So these neurons are not just involved in baroreflex, right? So this is the expected effect, but there's an additional effect in regulating the brain state. So that was neurons in the solitary tract. And then Yuan Yuan followed the downstream targets, right? So in this pathway, she activated the CVLM neurons or inactivated the CVLM neurons or inactivated the RVLM or activated the RVLM and also activated the AMB. So basically, I'm not showing you the data, but all of these experiments panned out, right? So anytime you do something that slow down the heart and decrease the blood pressure, you also promote non-REM sleep. Anytime you do the opposite, right? You increase the heart rate and increase the blood pressure, you also promote wakefulness. So basically, the conclusion that we had was that the cardiovascular baroreflex circuit, they have a daytime job, their primary job, which is cardiovascular control, but they also have a second job. So they moonlight in sleep control. Okay, so just to sum up this first part, right? The how question. So as I mentioned, falling asleep is about a change in the brain state, reduction of somatic and autonomic motor activity, right? Three kinds of things. So it kind of makes sense for the sleep control mechanism to inhibit all three systems, right? Brain arousal, autonomic, and somatic motor activity. Now, for the somatic motor system, I showed you the data on STN, as subthalamic nucleus, and SNR pathway. But we also have data showing that in the brainstem, there are these regions involved in motor control. They also promote sleep. Anytime that they suppress movement, they also promote sleep. Now, even though my lab, we did not look at the striatum, Zili Huang's lab and Michael Lazar's lab, they have looked at both the dorsal striatum and the ventral striatum, which is also called the nucleus accumbens. So in these striatal regions, the median spiny neurons that express the dopamine D2 receptors, if you activate those neurons, they also promote non-REM sleep. And of course, based on decades of work in the basal ganglia, we know that activation of those dopamine D2 receptor expressing striatal neurons can lead to the activation of neurons in the SNR through the so-called indirect pathway, right? So in other words, the multiple populations of non-REM promoting neurons, they're not independent, but in fact, they're interconnected with each other. In fact, the effect that they saw, Zili, Huang, and Michael Lazar's lab saw here could in fact be mediated by the sleep promoting effect that we saw in the SNR and the STN. And for the autonomic nervous system, I showed you the data in this part of the brain, right? The solitary tract, CVLM, and AMB. But we also have data in the midbrain, the periaqueductal gray, as I mentioned earlier, in the amygdala, and of course in the hypothalamus, and also Bill Wiston's lab who else? Several labs actually show that there are also neurons in the cortex that probably project to these autonomic subcortical regions. They also promote non-REM sleep. So given all these newly discovered non-REM promoting neurons in the forebrain, in the midbrain, and in the hindbrain for both the autonomic and the somatic motor circuits, it seems that this textbook model with a single sleep center in the POA is quite inadequate. Instead, sleep in the mammalian brain, non-REM sleep, is really controlled by highly distributed networks of neurons that deeply infiltrate both the somatic and autonomic motor circuits. So this is really the take-home message of the how question. Okay, so now we feel like we have a pretty good handle on the how question. Now, I'm pretty sure that there are still more non-REM neurons that we haven't discovered. In fact, when we wrote a review article on this several years ago, we just finished another review. We found there's so many more papers, and the good news is so far they still fit within this network, right? So I'm sure there's still more to be discovered. And yet, for me personally, I feel like, we sort of understand the basic logic of this network. So we've, in the past few years, switched our attention to the question of why we sleep. Now, we know that sleep deprivation causes a lot of problems, right? So in the brain, it causes problems with cognition, working memory, which you just heard about, memory consolidation, which is the main theme of this summer school, right? Of course, depression, maybe neurodegeneration, a lot of things in the brain. And beyond the brain, it also causes immunodeficiency, cardiovascular diseases, hormone imbalances, obesity, diabetes, and everything, right? So that means the sleep really interacts with many, many systems, not only in the brain, but also in the body. But in my mind, here is a big question. Does sleep support multiple independent processes, each for different function, right? Maybe some for brain functions, others for immune, others for metabolism. Or is there a single fundamental process that might underlie all these different effects, right? So my favorite example is feeding, right? So let's say just like sleep deprivation, if you do food deprivation, let's say I don't allow you to eat anything for 3 days completely. You can drink water, but you can't eat any food. I'm sure you would experience a lot of problems. It won't kill you. 3 days won't kill you, right? But I'm sure you would experience many metabolic problems and probably cognitive problems at all. I know for me personally, if I didn't eat today, I wouldn't be able to give this talk, right? I would be very cognitively impaired. And yet, we wouldn't say feeding is for cognition, right? We know that the fundamental purpose of feeding is the intake of energy and nutrients. So the question is, could there be something similarly basic for sleep, right? Even though sleep deprivation causes many problems, but maybe the underlying process is really pretty simple and fundamental. So instead of asking the original question of why we sleep, which I think is kind of poorly defined because different people can interpret that question differently, I decided to ask a separate question, which is there a single fundamental process underlying the multiple benefits of sleep? So we don't have the answer to that yet, but we have a hypothesis. I call it a catecholamine hypothesis, which is that inactivation of catecholamine signaling might underlie multiple functions of sleep. So catecholamines include dopamine, noradrenaline, and adrenaline. And in fact, these three molecules are biochemically related, right? So in the mammalian brain, in the body, in fact, we first synthesize dopamine using L-DOPA, and then we convert dopamine to noradrenaline, and then we convert noradrenaline into adrenaline. So I think that these are also functionally related, and inactivation of these might be sort of a fundamental process for sleep. So we focus on catecholamines for several reasons. First, it regulates not only brain arousal, but also somatic and autonomic nervous system, right? We all know that dopamine is so important for brain functions, it's super important for brain arousal. And of course, noradrenaline and adrenaline not only affect, for example, when you're running, we say you get your adrenaline going, right? So in fact, adrenaline is super important when you're in this sort of fight and flight behavior state, right? So activates not only somatic, but also autonomic motor activity. In fact, if you lose catecholamine signaling, I don't think you can be awake and conscious and behaviorally awake at all. Now, in addition to neurons and muscles, catecholamines also regulate metabolism and the immune system. And finally, we think that these neurons, their activity might link the functions of sleep and the drive for sleep, the homeostatic regulation of sleep. So in the remaining time, I want to tell you about two studies looking at how noradrenaline interacts with the immune system and the neuroendocrine system. So for the immune system, we first looked at microglia, which is the resident immune cells in the brain. Now, when there's disease and injury, we know that microglia switch to this defense mode, right? They look really angry. And so they're engaged in pathogen destruction, debris clearance, tissue repair, and so on and so forth. But in a normal healthy brain, hopefully for most of you in this room, their main job is to maintain brain homeostasis, right? So they have a different morphology and they basically just like try to keep the brain under control. Now, we know that one of the most important markers for homeostatic microglia is called P2Y12, which is a GPCR, G protein coupled receptor, that's activated by ATP and ADP released by neurons and astrocytes. So they kind of allow microglia through this receptor to kind of listen to the neurons and astrocytes and react accordingly to keep the brain under control. So the first experiment that we did was to manipulate P2Y12 signaling. So this was done by Chen Yan and Bing Li. So Bing is another former postdoc now running his own lab in China. So what we did was to cross this microglia-specific CRE-ER line with a GI dread reporter line. So we know that we know that the P2Y12 is a GI-coupled GPCR. So we use this genetic approach to activate GI signaling in the brain. So here is our experiment. This is a saline control. This is to use CNO to activate microglia GI. And here's a summary data. And you can see that compared to saline control, CNO activation of GI caused an increase in non-REM sleep and decrease in wakefulness. We also inactivated P2Y12 signaling by infusing a P2Y12 antagonist in the brain. So we think in the mammalian brain, this should only affect microglia directly because within the central nervous system, P2Y12 is specific to microglia. And this caused the opposite effect, right? So it caused a decrease in non-REM sleep and increase in wakefulness. So many of you probably know that the canonical effect of GI is a decrease of cyclic AMP, right? But in microglia, there's also a non-canonical pathway, which is to increase calcium. And people had demonstrated this in vitro previously. So we decided to look at microglia calcium in vivo. So we crossed this microglia-specific CRE-ER, GI dread, and cross the GCAM6 reporter line into it. So this is a triple transgenic mouse. And then we use 2-photon imaging. So here's a single microglia. And if you look at the RIs 1 through 5, they're all in the processes of microglia. And in general, we see more calcium transients in the processes compared to soma. And this is consistent with what people have seen previously, in particular, Longjun Wu's lab. Here's a summary compared to saline control. CNO activation of GI caused an increase in microglia calcium. The same thing is true if we infuse the P2Y12 agonist. So it seems that P2Y12/GI activation caused an increase in calcium. Now, we also know that P2Y12 causes an increase in sleep. So the question is, what's the relationship between microglia calcium and sleep? So for that, we just went to the imaging sessions without any drug, just the saline control, just to correlate microglia calcium with brain state. So in general, we see more calcium transients during non-REM sleep compared to wakefulness. And this is the transition. You can see the calcium goes down when the animal wakes up, and it goes up when the animal falls into non-REM sleep. So it seems that microglia calcium is correlated with natural sleep. But then the question is it causal? So we think that microglia calcium might be causally linked to sleep for two reasons. First, when we expressed this thing called the P130pH, which is IP3 sponge, it basically sucks up the IP3, which is important for internal release of calcium in microglia. When we use that, we can see that the increase in microglia calcium induced by GI is largely gone, right? So this magenta line is without the sponge, and this blue line is with the sponge. So you can see that the increase in calcium is largely gone. And the increase in non-REM sleep at the behavioral level is also largely blocked. In contrast, when we activated GQ, right, the canonical effect is to increase microglia calcium, we still saw a robust increase in non-REM sleep. So this is a loss of function, right? Block the calcium increase. This is gain of function, increase calcium through a different pathway. So in both cases, we saw the change in non-REM sleep. So we think that perhaps calcium does cause sleep. But then the question is how, right? So microglia are not even neurons. And in order to induce sleep, you have to affect neurons somehow. So next, we looked at the interaction between microglia and noradrenaline. So we know that neurons in the locus coeruleus release noradrenaline all over the brain. And we know that neurons, astrocytes, and microglia all express a ton of adrenergic receptors, right? So there are multiple adrenergic receptors, and microglia express a ton of them. So next, we wanted to look at the interaction between microglia and noradrenaline. The first thing we did was to use the sensor of noradrenaline developed by Yulong Li's lab. So we express the sensor in both the locus coeruleus and in the prefrontal cortex. And here's our imaging session. And basically, noradrenaline level is higher during wakefulness than non-REM sleep. Now, this is extremely old news because people have known for 40 years that the locus coeruleus neurons are more active during wakefulness. So you see more noradrenaline, totally expected. But the important thing here is that the change in noradrenaline and the change in microglia calcium happen in opposite directions, right? So when noradrenaline goes up, microglia calcium goes down, and vice versa. So next, we wanted to look at what happens if we activate microglia GI and measuring NE level. And in fact, that caused the decrease in the NE level compared to saline control. So they're not just anti-correlated. In fact, microglia calcium can cause a decrease in NE. Now, we know that P2Y12 not only allows microglia to sense ATP and ADP in the brain, right? So they're ATP receptors. We know that microglia, through the work of Ann Schaefer, microglia can also help to metabolize ATP and ADP into adenosine in the brain. And we also know for a long time that adenosine is a very powerful somnogen that can promote sleep. So next, we used another one of Yulong Li sensors to sense adenosine. And we found that microglia GI activation using CNO caused a large increase in adenosine concentration. And when we infused an adenosine receptor agonist, we saw a large decrease in the NE level. So we think that based on this set of experiments that the decrease in the NE caused by microglia GI activation is partly mediated by through adenosine. So basically this pathway is shown here. So now we've looked at how microglia affects noradrenaline. What about interaction in the opposite direction? So next we went back to microglia calcium imaging. And when we infused noradrenergic receptor antagonists, right, either a cocktail that blocks a range of receptors or a beta-2 adrenergic receptor antagonist, right? So this is a specific blocker. In both cases, we saw a large increase in microglia calcium. So basically, NE also inhibits microglia calcium, and it seems that the interaction between microglia and noradrenaline are mutually antagonistic. Now we know that microglia calcium is important for their motility and phagocytotic activity. So microglia, like I said, keeps the brain under control. And a lot of that is to kind of move around and try to, clear up damaged neurons and debris through phagocytosis. And calcium plays a very important role in that. So this inhibition by noradrenaline indicates that if you do sleep deprivation and keep noradrenaline level high, right? So we know noradrenaline is high during wakefulness. So if you keep noradrenaline level high for a prolonged period of time during sleep deprivation, then this pathway can perhaps interfere with the microglia function shown here, right? But what's interesting here is that microglia can also signal back. We think that perhaps when microglia sense the needs for these housekeeping functions in the brain, they might, through this pathway, inhibit noradrenaline and therefore tell the brain to go to sleep so that the microglia can then perform these housekeeping functions. Okay, so this is the story on the interaction with the immune system. And now I'm going to quickly take you through another study on how sleep interacts with the neuroendocrine system through noradrenaline. So we looked at the growth hormone, which is important for tissue growth, metabolism, neurogenesis, and neuroprotection, not only during development, but also in adults as well. So we know that even adult growth hormone deficiency causes obesity because it's important for growing lean muscles, right, and burn fat. And growth hormone is mostly released during sleep. So this is some human data. And you can see that during the nighttime sleep, there's a surge of growth hormone. This is not just circadian because if you sleep deprive the subject in the second night, then you don't see the surge. But then if you allow them to go to sleep during the day, then you see the surge again, right? So growth hormone release is sleep dependent. And when we measure growth hormone in mice, we also saw similar things. So growth hormone during both non-REM sleep and REM sleep is higher than during wakefulness. So how does sleep control growth hormone release? Now we know that growth hormone release is strongly regulated by a population of neurons in the arcuate nucleus in the hypothalamus. So these neurons release growth hormone releasing hormone, GHRH. So they release this peptide into the median eminence. And when the peptide reaches the anterior pituitary, it stimulates the production and release of growth hormone, which is then carried by blood to the rest of the body to function. So the first thing that Xin Lu, a postdoc in my lab at Berkeley, did was to characterize the relationship between GHRH neuron activity and growth hormone release. So she expressed channelrhodopsin in GHRH neurons, and then and then collected blood and used ELISA to measure growth hormone in blood. So here she stimulated GHRH neurons for 2 minutes per trial, but varied the frequency. The higher the frequency, the higher the GH level. And here she kept the frequency to 10 hertz, but varied the duration. The longer she stimulated, the higher the GH. So everything checks out pretty cleanly. So next, she used calcium imaging to measure the GHRH neuron calcium activity during natural sleep-wake states. So here is an example session, and here's a summary. So generally speaking, you can see that GHRH neuron activity drops when the animal wakes up from non-REM sleep. It increases when he or she goes into non-REM sleep from wakefulness. There's a further increase from non-REM to REM sleep. And then again, when they wake up from REM sleep, there's a drop. So she also, in addition to calcium imaging, she also tagged these neurons with channelrhodopsin and then used output recording. And she found similar changes across brain states. So we think that the higher activity of GHRH neurons during sleep is one explanation for why GH level is higher during sleep. But in fact, there's another level of regulation. So when she fixed the stimulation frequency and duration to 5 hertz for 40 seconds, what she found is that when the trained lasers fall during wakefulness or non-REM sleep or REM sleep, the same laser stimulation caused a higher GH release during sleep than during wakefulness. In other words, the same activation is more effective during sleep in inducing GH release. So this was also confirmed with chemogenetic activation. So here she used GQ to activate the neurons, and she can see a prolonged large increase in GH release. But if she applies sleep deprivation right after CNO injection, there's still an increase in GH, but you can see that compared to control, this increase in GH release is cut down by more than half, right? So meaning that sleep during this period is still important for boosting a growth hormone release. Okay, so in addition to GHRH, which is a positive regulator, we also know that there's a negative regulator of GH release, which is, in this case, the somatostatin neurons. So there's a population in the periventricular nucleus, PEV. So here we use retrograde tracing and anterograde tracing to show that the PEV somatostatin neurons project strongly to the median eminence, which is where the GHRH neurons project to, right? So the positive and negative regulators convert here to have the opposite effects on growth hormone production and release. And here, when Xin Lu chemogenetically inhibited the SST neurons, she saw a large increase in growth hormone release, right? So this indicates that indeed these guys are negatively regulating GH release. So next, she imaged the natural activity of the SST neurons. So in this case, she used the SST FLIPO and the GHRH Cree, a double transgenic mice, to simultaneously label the SST and GHRH neurons. So here's a simultaneous imaging. Black is GHRH and magenta is SST. And here's a summary. So generally speaking, during wakefulness, right, the magenta line, SST neurons, have higher activity than the GHRH neurons. So that's true here and also here. But when the animal is in either non-REM sleep or REM sleep, it's the other way around. The GHRH neurons have a relatively higher activity. So we think that this is why GH release is higher during both non-REM and REM sleep. Okay, so we know that GHRH has a lot of effects in the body, but it also has some effects in the brain. So to get a better idea of what growth hormone does in the brain, we first looked at the expression of the GH receptors. So we found a lot of GH receptors in these regions. So these are all in the hypothalamus. And these are very well known to be engaged in negative feedback, right? So the idea is that when these receptors sense a high level of growth hormones in the circulation, in the blood, then they inhibit the activity of GHRH neurons and also the synthesis and release of growth hormone. So these are all very well known. But to our surprise, we also found growth hormone receptor expression in the locus coeruleus, right? So here's tyrosine hydroxylase is a marker for the noradrenergic neurons, and you can see very high overlap with growth hormone receptors. So next, we wanted to look at what the effect of GH is on LC neurons. So when Xinlu infused mouse growth hormone into the brain, she saw, compared to saline control, she saw a large increase in false expression in the locus coeruleus neurons. And our collaborator, Fujun Huang, in Junding's lab at Stanford, helped us to make recordings in brain slices of LC neurons. And she found that growth hormone application caused an increase in the excitability of these LC neurons. To test whether these are caused by the growth hormone receptors expressed in the LC neurons, Xinlu used CRISPR-Cas9 to target the growth hormone receptors to knock them down specifically in the locus coeruleus neurons. And here you can see that compared to control. So this is just a random sequence, right? Doesn't knock out the receptors, but we still inject the virus. Then you still see the C-false expression. But when we knock down the growth hormone receptors, the C-false increase is greatly reduced. And the same thing is true for the excitability increase that we saw in brain slices. So now we no longer see the increase in excitability. So to sum up this part, basically we found two ways in which sleep can promote growth hormone release. One is to increase the GHRH neuron activity relative to the somatostatin neurons, and the other is to make the GHRH neuron activity more effective in inducing GH release. But what's interesting is that GH can also feed back to the circuit, right? So when there's a high level of GH in the circulation, it can increase the excitability of the noradrenergic neurons, which then promotes wakefulness and suppresses sleep. So if the function of sleep, one function, is to promote tissue growth through growth hormone release, then this feedback pathway would allow the brain to know when and how much sleep is needed in order to perform this function. Okay, so finally, I just want to spend maybe 2 minutes to tell you one more experiment, which is incredibly simple, right? So we wanted to see whether the release of noradrenaline by locus coeruleus neurons is affected by sleep. So this is a very simple experiment done by Jiao in my lab. So she expressed crimson R, which is a red opsin in the locus coeruleus neurons, and then GRAB-NE in both the locus coeruleus and the prefrontal cortex. And when she applied a brief laser pulse to activate the LC neurons, you can see a transient increase in noradrenaline. So I know that this summer school is about, synaptic plasticity. So you all know about EPSP, right? LTP, you see evoked EPSC. So here you can think of something like EPSC, except that it's a neuromodulator rather than a neurotransmitter, right? So it's a measure of synaptic strength for noradrenergic neurons. Now, this is the control, and then she applies 3 hours of sleep deprivation. And then you can see that right after the deprivation, the evoked NE release, right? See these little transients, is greatly reduced compared to control. But then after you allow the mouse to sleep for, let's say, an hour or so, then it partially recovers. And here's the summary, right? So the black is control without sleep deprivation. Now you see this gradual decrease, and this is unfortunately a reflection of bleaching, right? So GRAB-NE is a fluorescent-based sensor, and anytime you do these long-term imaging experiments, you have bleaching, right? But if you compare sleep deprivation with the control, you can see that right after sleep deprivation, there's a large decrease, and then gradually it catches up. So this plot shows the ratio between SD and the control curve. And you can see that this immediate reduction right after 3 hours of sleep deprivation recovers over about 40 minutes of recovery sleep. So this was true in both the locus coeruleus region and also in the medial prefrontal cortex. So next, the question is, what happens during sleep deprivation? Now, we know that the noradrenergic neurons are more active during wakefulness. So maybe the drop in noradrenaline release is just because they've been active for too long, right? You don't allow them to rest, right? So to test that, Jiao replaced the 3 hours of sleep deprivation with a relatively high level of optogenetic activation of the LC neurons. And that pretty much caused the same effect, right? As compared to control, there's immediate drop, but after you stop this stimulation and go back to low frequency testing, you can see the recovery over half an hour, 40 minutes or so. So we think that the sleep deprivation induced decrease and subsequent recovery of noradrenergic transmission is partly explained by the fact that these neurons, during sleep deprivation, they're just tired, right? They've been active for too long. Okay, so just to sum up everything I said today, basically we looked at noradrenergic interaction with microglia, right, in the immune system, also with the growth hormone secretion pathway. So the point here is that noradrenaline and other catecholamines not only regulate metabolism and the immune system, they can also integrate signals from the immune system and the endocrine system. I mean, like this pathway and that pathway, right? So this feedback allows them to perhaps figure out what the body needs, right? And then link the functional brain to sleep need. In addition, these neurons also experience activity-induced functional fatigue. By fatigue, I mean their ability to release noradrenaline is reduced, right? And so we know that catecholamine neurons are particularly vulnerable to neurodegeneration. For example, we know that dopamine neurons are the first to die in Parkinson's disease, and noradrenergic neurons die early in both Parkinson's disease and Alzheimer's disease, right? So these neurons, just because catecholamine metabolism per se generates a lot of toxic, reactive oxygen species that are damaging to neurons. So we think that this kind of vulnerability means that these neurons are intrinsically, they're just like fragile. After being active for a while, they just need to shut down to rest for a while. And we think that the recurring need for rest and recovery could provide another link between the function of sleep and our need for sleep. Basically, you get tired, sleepy. So basically, based on all of these, we think that the catecholamine hypothesis is quite plausible that catecholamine inactivation might be a basic reason for many functional benefits of sleep. So that's it. And I think I mentioned the people in my lab who did the work along the way. I won't repeat their names. We also have many collaborators who helped us with the various techniques. And I guess I have a couple more minutes for questions in case you're not too hungry yet.